For centuries, health was defined by the balance of fluids, known as humors. Early Greeks like Hippocrates posited that a person's personality and susceptibility to fever were governed by substances like black bile. This conclusion was reached through reflective thought rather than physical observation. Parallel systems, such as Indian Ayurveda, utilized similar conceptual frameworks to categorize the body's internal state. While complex, these ancient models lacked an empirical method to verify how the body actually functioned. This philosophical approach was challenged by the arrival of the experimental method. William Harvey used physical evidence to map the actual circulation of human blood, shifting the focus from abstract humors to measurable mechanics. The invention of the thermometer provided further contradiction to the humor theory. By using it to show that individuals supposedly possessing black bile maintained a normal body temperature, researchers demonstrated that the hypothesized link between these humors and fever did not exist. These discoveries moved the definition of health away from philosophical balance and toward a biological reality that could be measured and tested. Modern medicine defines health as a state of physical, mental, and social well-being. This standard views health as a contributor to productivity and longevity, looking beyond the simple absence of a clinical disease. Our primary defense is the immune system. It functions as a complex network continuously influenced by our neural and endocrine systems to maintain the body's internal stability. This baseline is subject to internal disruptors, such as genetic deficiencies or defects that a child inherits from their parents at birth. External factors also play a role, including lifestyle choices regarding diet, water quality, and the balance of rest and exercise we provide our bodies. The most dynamic threats are infectious pathogens. These organisms invade the host to multiply and interfere with vital activities, often resulting in functional or structural damage to the body. To protect the body, we must map how these foreign organisms exploit human anatomy to survive and spread. Successful infection depends on adaptation. A pathogen must be able to survive within the specific, often hostile environment of its host. The bacterium Salmonella typhi illustrates this. After entering through contaminated food or water, it must survive the low pH of stomach acid and resist digestive enzymes to reach the small intestine and enter the bloodstream. The resulting typhoid fever causes sustained high temperatures, weakness, and stomach pain. In severe cases, it can lead to intestinal perforation, with the presence of the bacteria confirmed through a Widal test. Other bacteria target different systems entirely. Streptococcus pneumoniae and Haemophilus influenzae infect the air-filled sacs of the lungs, known as alveoli. As the infection progresses, the alveoli fill with fluid, hindering respiration. This can cause the lips and fingernails to turn blue as oxygen levels in the tissue drop. Viruses show similar anatomical specificity. Rhinoviruses target the nose and respiratory passage. While highly infectious, they remain localized in the upper respiratory tract, causing congestion and sore throats without migrating into the deep lungs. These pathogens succeed by identifying and exploiting very specific microscopic niches within our anatomy. Some organisms, like the protozoan Plasmodium, rely on a more complex strategy. To complete its life cycle and cause malaria, it requires two distinct hosts. The cycle begins when an infected female Anopheles mosquito bites a human, injecting the infectious parasite known as sporozoates. These parasites travel to the liver, multiplying within the cells before attacking the red blood cells. Once inside the red blood cells, the parasites continue multiplying until the cells rupture, releasing new parasites. This rupture also releases hemozoin, a toxic substance. Its presence in the blood directly triggers the recurring chills and high fevers of malaria. The loop continues when another female mosquito bites the infected person, ingesting the parasites. Inside the mosquito, they develop into new sporozoates and migrate to the salivary glands, waiting to be introduced to a new human host. Through this biological loop, Plasmodium leverages the systems of two different species to ensure its own survival. Beyond microbes, humans are also targeted by macroscopic parasites like helminth worms. Filarial worms, such as Wuchereria, can live inside human lymphatic vessels for years, slowly causing chronic inflammation. This inflammation leads to the gross deformations of the lower limbs and genital organs, known as elephantiasis. These pathogens are typically introduced to the body through the bite of a mosquito vector. Other pathogens are purely superficial. Fungi from the genus Microsporum are a common cause of ringworm in humans. These fungi thrive in the heat and moisture found in skin folds, such as the groin or between the toes, where they grow across the surface of the skin. The result is intense itching and dry, scaly lesions. Because they reside on the skin, these fungi are easily spread through contaminated soil or shared items like towels and combs. Pathogens have adapted to survive in environments across the body, from the deep lymphatic system to the epidermal layers. Effective epidemiological defenses are built upon the biological profile of the target pathogen. When we understand how an organism travels, we can identify the most effective ways to stop it. Pathogens that infect the gut, such as those causing typhoid or amoebiasis, generally spread through contaminated food and water. For these, the primary defense is waste disposal and the protection of water resources. Airborne diseases like pneumonia or the common cold require a different approach. Because they spread through droplets and contaminated objects, defense involves maintaining distance from infected individuals and disinfecting shared surfaces. However, these hygiene measures have little effect on vector-borne diseases like malaria or dengue, where the pathogen is delivered directly into the blood. Breaking this cycle requires controlling the vector. This involves eliminating stagnant water, using mosquito nets, and introducing predators like Gambusia fish to consume mosquito larvae. Interventions are most effective when they are designed to intercept a pathogen's specific transmission route. Immunology and biotechnology have provided the tools to dismantle these transmission loops more permanently. Vaccination and immunization programs represent some of our greatest successes. These large-scale efforts enabled the total eradication of smallpox. Similar programs have brought diseases like polio, diphtheria, and tetanus under control, replacing natural susceptibility with artificial resistance. For active infections, the discovery of antibiotics and other drugs has allowed us to treat diseases by targeting the biological machinery of the pathogens themselves. Because the landscape of disease is always changing, biotechnology continues to develop newer and safer vaccines to address emerging threats. Human health is a continuous biological defense, sustained by our scientific understanding of these microscopic adversaries.